The phone app suggested that this fern, with its one pinnate pinnatified blade, round sorry, and reniform indusia, was Thelypteridaceae interrupta. But as I researched this name, I found that the same plants went by various other names, such as Cyclosorus interruptus and Dryopteris interrupta. The similarities within various ferns led to treating them as a single family, Dryopteridaceae. But further characterization placed some in the family Dryopteridaceae and others in the Thelypteridaceae. The sample plant observed is Thelypteridaceae due to its two vascular bundles in the cross-section of the basal part of the stipe and unicellular hairs on the stipe, lamina, and veins. Species within the Dryopteridaceae family would have 5 to 7 vascular bundles organized in a U shape in the petiole cross-section and generally be scaly, not hairy. An alternate taxonomic treatment refers to the plant Cyclosorus interruptus as Thelypteridaceae interrupta in the family Asplanaceae. However, I shall follow the taxonomic treatment supported by Fawcett and Smith in 2021 and refer to the family as Thelypteridaceae. The similar appearance of ferns in the family Thelypteridaceae also causes difficulties in their taxonomic assignment at the genus level. For example, Fawcett and Smith in 2021 supported using the name Christella parasitica rather than Cyclosorus parasiticus or Thelypteris parasitica. These synonyms in plant names arise because the earlier author who described the plant placed it in a particular taxon, but subsequent authors moved it to a new taxon which they believe reflects a more accurate classification. Another reason the same plant is named differently is that different people may have named the same plant species independently and unknowingly under other names. The correct name will be the oldest validly published name. With diverse publications worldwide, it is not easy to determine. Assignment of the species to the genus Cyclosorus was confirmed as the sample plant has the combination of characteristics recorded in Holtham's publications to characterize members of the Cyclosorus genus. That is, it has rhizomes that are long creeping, nearly scaleless, blades truncate at the base without greatly reduced proximal pinnae, anastomosing veins, usually one pair, costa bearing persistent flat ovate scales abaxially, the presence of long-stalked glands on the sporangial stalks and receptacle, as well as sessile orangish or reddish glands on the lamina. To check further on the identification of the sample as Cyclosorus interruptus, I compared the sample to some other similar-looking plants or plants referred to as Cyclosorus by other botanists and found in Malaysia. With 1,200 Thelypteridaceae, it is not a comprehensive list of comparisons. The sample differed from Christella dentata, otherwise known as Thelypteris dentata, as the sample has scales on the lower surface of the frond and costa, while the upper surface is virtually hairless, pinnae lobed less than halfway to the costa, and no sori on the veins that unite below the sinus. The sample differed from Christella parasitica, otherwise known as Thelypteris parasitica, as Christella parasitica has proximal 1 or 2 pairs of pinnae reflexed, deep lobes on the pinnae segments from 2/3 to 3/4 way towards the costa, and absence of sori on the lobes. The sample was easily differentiated from Christella aridas or Thelypteris arida by the sample's basal pinnae hardly being shortened and the pinnae lobes being triangular obtuse to triangular cuspidate, instead of the proximal pinnae being shortened and the pinnae lobes being oblong subdeltoid, as in Christella aridas. The sample differed from Christella subpubescens or Thelypteris subpubescens. As in Christella subpubescens, the lower pinnae is sometimes reduced to mere auricles and the terminal pinnae is long, narrow, and triangular at the apex. Christella subpubescens also has basal 2 to 3 pairs of veins anastomosing and pinnae segments round at the apex, unlike the sample which only has a single pair of veins anastomosing and pinnae lobes triangular obtuse to triangular cuspidate. The sample distinctly differed from Chingia ferox, also known as Thelypteris ferox, as the sample does not have the terate-type scales or the ex-induciate sori close to the costules. So the sample was most probably Cyclosorus interruptus of the family Thelypteridaceae from the following: frond blade pinnate pinnatified with the basal pinnae hardly shortened, Type yellow-brown to almost black at the base, slightly polished in appearance and nearly hairless, though it may be scaly near the base. The rhizome underground and widely creeping. Pinnae not deeply lobed, lobes triangular obtuse to triangular cuspidate. A single pair of veins from adjacent lobes anastomose below the sinus. The excurrent vein terminates in the sinus between the lobes. A second lobe vein terminates in the sinus or just above, and free veins end in the lobe margin. Sori are circular and medial on the veins and absent on the veins that unite below the sinus. Indusia are reniform with unicellular acular hairs. Fronds are abaxially densely set with hyaline unicellular acular and oblong hairs, but adaxially virtually hairless. Spherical glandular cells are present on the lower surface of the frond and long hairs with terminal red glands on sporangial stalks. For further exploration of this confusing family, here are some references. Do subscribe to the channel. The strange greenery around you may become more identifiable. Thank you for watching.